The laboratory methods include culture, rapid detection, and serologic test. Culture. The viruses can be cultivated in animals, in fertile eggs, and in cell cultures. Specific types of tissue culture cells are used to grow viruses. Rapid detection includes electron microscopy for the detection of viral particles, detection of viral antigen by enzyme-linked immunosorbent assay, ELISA, and immunofluorescence, IF, and the detection of viral nucleic acid by polymerase chain reaction, PCR. Serologic test is to detect the immune responses to the specific virus, such as specific IgG or IgM. Serologic tests are used for the identification of viruses that are difficult to isolate and grow in cell culture, as well as viruses that cause diseases of long duration. For example, EB virus, HBV, and HIV. The last question is, how does our immune system fight the viruses? One is interferon, IFN. Non-specific host defense mechanisms are usually elicited very soon after viral infection. After the virus penetrates the natural barriers, such as skin, it activates the innate host defenses. For example, fever, interferon, macrophages, dendritic cells, natural killer cells. The innate immune responses attempt to limit and control local viral replication and spread. The most prominent among the innate immune responses is the induction of interferons. Interferons are host-coded proteins that are members of the large cytokine family and which inhibit viral replication. When a virus invades host cell 1, the infected cell makes interferon. Interferon induces changes that protect host cell 2. Interferon stimulates cell to turn on genes for antiviral proteins production. The other one is a natural killer cell. Natural killer cells are an early antigen nonspecific component of the immune response to viruses. Let's see the diagram. Just after the exposure to the viruses, the interferon can be produced. Then the natural killer cells increase and kill the infected cells. And then specific cytotoxic T cell, CTL, kill the infected cells. Innate responses prevent most viral infections from causing diseases. Specific humoral and cell-mediated immunity to viruses. Virus-encoded proteins serve as targets for the immune response. You should note that 1. Virus-infected cells could be lysed by CTL. 2. Humoral immunity protects the host against reinfection by the same virus. 3. Neutralizing antibody directed against the surface proteins blocks the initiation of viral infection. 4. Secretory IgA is important in protecting against infection by viruses through the respiratory or gastrointestinal tracts. Serologic tests are used to measure the immune response. An elevated titer of a specific IgM suggests a recent primary infection. Fourfold increase of IgG titer between the acute phase and the convalescent phase is of diagnostic value. Keywords of this part are vertical-horizontal transmission, CPE, inclusion body, inapparent, acute, chronic, latent, slow infection, and interferon. A quiz about interferon. Interferons are an important part of the host defense against viral infections. What is interferon's principal mode of action? A. It protects the virus-infected cell. B. It is present in a serum of healthy individuals, providing a viral surveillance role. C. It coats viral particles and blocks their attachment to cells. D. It induces synthesis of one or more cellular proteins that protect the cells from infection of viruses. Which one is your choice? Yes, D is correct. Interferon cannot protect the infected cells, but protect the neighbor cells. Summary of this chapter. We have discussed about the structure, replication, variation, and classification of the viruses, and viral infection and host response, and laboratory diagnosis of viral infection. Now, could you answer all these questions? What are viruses? They are the smallest pathogens and contain only DNA or RNA. The nucleic acid is encased in a capsid and may be surrounded by an envelope. What is the structure of a virus? Basic structure, core, and capsid. Some have enveloped and spikes. How do viruses replicate? Self-replication, 6 steps: attachment, penetration, uncoating, biosynthesis, assembly, and release. How do viruses change their antigenicity? They do mutation, gene recombination and gene reassortment, and phenotypic mixing. How to classify the viruses? The viruses can be classified based on biological properties and the transmission route, and so on. How does viral infection manifest itself? Please remember the iceberg concept. How can viral diseases be transmitted? Vertical transmission and horizontal transmissions. How do we confirm the diagnosis by lab test, culture, rapid detection, and serologic test? How does our immune system fight the viruses? Interferon, natural killer cells, and specific immune responses. Hope you can answer all of them and enjoy the learning process. All these informations will come again when we learn about the different viruses. Thank you for your listening. See you next class.